This lesson provides an overview of the structure and function of neurons, and here are the objectives. Here is a generic diagram of a neuron. Now, neurons come in many different kinds of shapes and sizes. They don't all look quite like this, but this one will serve as an overview, and the others are just variations on this theme. So neurons are cells. They have a cell body, and another name for the cell body of a neuron is the soma. That's another name you'll sometimes hear. And of course, they have a nucleus, being eukaryotic cells. These many projections here leading into the cell body are called dendrites, and they receive signals from other neurons. Whereas this single long projection that goes this way is called the axon. This is the axon going here, and it transmits the signal to the next neuron. Now, the nature of the signal within the neuron is electrical by movement of ions, and the signal that goes down the axon is called the action potential. We'll look at that in more detail in future lessons. The action potential is the electrical signal that travels down the axon. When it gets to the end, it reaches the axon terminal, and if I draw this entire neuron here, just like this lollipop shape, so I've replaced this whole thing like that, then it's here at the axon terminal that that signal gets transmitted to the next neuron, and we'll look below at how that happens. Now, this makes it look like one neuron connects to one neuron, connects to one neuron, but many neurons can connect to one neuron, and this one here, for example, will integrate the signal from all of them. So this is the axon terminal. Again, we'll look at that in more detail below. Wrapped around the axon are many Schwann cells, and the Schwann cells make up the myelin sheath. If we look at this neuron end on, this is the neuron from the end, and then wrapped around it we have these Schwann cells, and their plasma membranes make up a structure called the myelin sheath. This myelin sheath is a form of insulation that prevents that electrical signal from leaking out and makes the action potential travel down the axon much faster. These interruptions that you see here and here and here in the myelin sheath between the Schwann cells are called nodes of Ranvier. And the reason this whole setup speeds the transmission of the action potential is that it jumps from one node of Ranvier to the next. It doesn't need to be transmitted through these sections that are insulated by the myelin sheath. So the action potential jumps from one node of Ranvier to the next. This little squiggle that I'm drawing is meant to represent that electrical signal of the action potential. And this jumping conduction from one node of Ranvier to the next is called saltatory conduction. Saltatory means jumping in the same way that sauter in French means to jump, or somersault. It has to do with flipping. So saltatory conduction is the term that we use for this jumping manner of transmission from one node of Ranvier to the next. So myelination is one way that the speed of action potentials is increased. Another way that the speed of action potential transmission can be increased is through making the axons have a larger diameter. So if we look end on at two different axons, one like that and one like that, this one will transmit signals, action potentials, faster. And in fact, squid, which don't have the ability to myelinate their neurons, have very large axons called giant axons that are responsible for the jet propulsion reflex that they use to get away from predators. That needs to be a fast reflex, so they use very large diameter neurons. Or large diameter axons. So myelination and having a large axon diameter are two different ways that speed of transmission can be increased. So I've said that the nature of the signal traveling within the neuron is electrical, but what about here at the junction between one neuron and the next? This area here, the junction, is called the synapse, and the signal in between one neuron and the next is actually chemical. Let's look now at how that happens. So we call this synaptic transmission because this whole junction of two neurons is called the synapse. Note, by the way, that we've turned the diagram above to the side. So now we're looking like this, and we're zooming in on this part here, where the axon, this is the axon of one neuron, is terminating on, and I haven't drawn the dendrites here, but this would actually be not the cell body, but a dendrite of the following neuron. This whole structure, this whole junction with two neurons and a gap in between, is called the synapse. The space itself is called the synaptic cleft. And we call the neuron that is leading into the synapse the presynaptic neuron. The presynaptic neuron. And the one that is receiving the signal and coming out of the synapse, the postsynaptic neuron. And that's, of course, only relative to this particular synapse. The postsynaptic neuron here, shown in purple, is going to be the presynaptic neuron for the next synapse that's down this way. So we said that the signal that's traveling down the axon, the action potential, is an electrical signal. But when that electrical signal of the action potential reaches the terminal, it causes an influx of calcium ions through ion channels, and those calcium ions actually serve as a trigger for exocytosis of vesicles containing neurotransmitter. So these red dots are neurotransmitter. It's all one word, I'm just hyphenating it here because I've run out of space. That neurotransmitter could be something like dopamine, serotonin, etc. And the neurotransmitter is then released by exocytosis into the synaptic cleft, where it then binds to transmembrane receptors on the postsynaptic neuron and triggers an action in that postsynaptic neuron. As an example, the neurotransmitter acetylcholine, that's another neurotransmitter, often abbreviated as capital A, capital C, lowercase h, can bind to a receptor called a nicotinic acetylcholine receptor. And it's called nicotinic because in addition to acetylcholine, it'll also bind and respond to nicotine. And when acetylcholine binds to the nicotinic acetylcholine receptor, that receptor is actually a ligand-gated ion channel, meaning it opens in response to the binding of a ligand, in this case the acetylcholine, and that allows positive ions, cations, into the postsynaptic neuron, which can then cause transmission of an electrical signal in that postsynaptic neuron as well. So to get past this gap here, neurotransmitter, a chemical signal, is used before an electrical signal can be restarted in the postsynaptic neuron through a mechanism like this. We'll be able to look at action potentials and synaptic transmission in more detail in future lessons, but this lesson has provided an overview of the structure and function of neurons.